For those of you who are not familiar with the Critical Path Institute, CPATH is it's a nonprofit that works within the pre-competitive space, typically to address drug development challenges through consortia. Now, these consortia include multiple different partners from the pharmaceutical industry, regulatory authorities, academic institutions, other nonprofits, and funding institutions. And the idea is that these consortia bring together data and expertise that then can be used to develop solutions for the drug development challenges. Oftentimes, these solutions are in the form of drug development tools, and that's the case of what I'm presenting today, which is a set of drug development tools that we're working on in the Modeling and Simulation Working Group in order to help improve cardiac safety assessment for drugs for TB. But also, these algorithms that I'm presenting today can also be used for multiple therapeutic areas. I'm not going to talk much about CPTR because there are many here who could probably speak better to the work that CPTR is doing in TB. However, I will point out that the cardiac testing paradigm that I'm going to be showing is really addressing the two bullet points that are bolded there, which is to improve regimen selection for TB as well as to accelerate the development of these regimens. And by informing cardiac risk assessment, hopefully we can meet these two objectives. So, stepping back and framing the actual challenge that we're trying to address with this work. Now, this is not specific to TB drug development. This is of interest to new chemical entity development throughout a variety of therapeutic areas. In that drug-induced torsade DuPont, TDP, is often a concern for drug developers and is often screened for by screening against hERG inhibition and clinically by looking for QTc prolongation, so prolongation of the corrected QT interval. Now, there's a challenge here, as in recent years, the QT interval has been called into question, that it may not be the best biomarker for predicting the likelihood of torsade DuPont clinically. That's because this is a multifactorial effect. There are questions related to the influence of the hERG ion channel inhibition versus other ion channel inhibition, changes in the QT interval versus potentially other ECG changes that may be of interest and may be better biomarkers of torsadogenic potential. Also, early in development, a question around what is a therapeutic dose versus a supratherapeutic dose. This is not always well defined early on, so what dose range may potentially precipitate a torsadogenic effect is not necessarily clear. And in the case of TB, obviously we're concerned with combining multiple agents together in the form of a regimen. Now, there are several anti-TB drugs that are associated with QT prolongation. We heard today about bedaquiline in particular. Moxifloxacin is a great example and is arguably the most widely studied drug in terms of QT prolongation, and that is the positive control used in TQT trials. So when you start combining drugs with multiple effects on cardiac ion channels into a regimen, there are questions obviously with regard to the likelihood of causing torsades in the clinic. So what the Modeling and Simulation Working Group is trying to do to address these uncertainties is to really put together a quantitative systems toxicity testing framework for cardiac risk assessment. This spans the development paradigm from early discovery all the way through late phase assessment in the clinic, and is intended to leverage available data at each phase in order to convert that data into a clinically relevant classification for torsades risk. Today I'm going to focus the bulk of the talk on the early discovery portion, but near the end I will talk a bit about what we're doing for the later stage assessment. So the early discovery screening component of this platform is really designed to leverage in vitro ion channel inhibition data or even predicted ion channel inhibition data through the use of Simcyp's cardiac safety simulator, which is a virtual cardiac myocyte or string of cardiac myocytes, in order to generate outputs such as a pseudo ECG, which you can view as a virtual ECG, in order to generate metrics that could be used to predict whether or not a drug, on the basis of ion channel inhibition data only, is likely to be torsadogenic when used in the clinic. And the idea here is that this algorithm could be used to prioritize compounds. So if you have a set of lead compounds or potential compounds and you want to pick a candidate drug, if you want to rank these compounds in terms of their likelihood of causing torsades, this algorithm could be used, perhaps to bin them into low, moderate, and high risk categories. Additionally, it could be used to inform your preclinical testing strategy, perhaps when you would run your safety pharmacology studies for cardiac risk assessment. If you have a drug that may have a higher potential risk, you may want to push those studies up further or maybe run more studies to help inform that risk. Also, with first-in-human dosing, if you have a compound that is projected to have a potentially high risk of torsades, perhaps you may opt to have a greater safety margin with your first-in-human dosing or include more thorough cardiac monitoring, for example, digital Holter monitoring in your first-in-human study. So getting to the actual algorithm itself, this was developed, as I mentioned, based on the cardiac safety simulator from SimCip. Now what was done was that 96 compounds were identified that are clinically used that have known classification of being either torsades positive or torsades negative. And this is based on the Credible Meds QT drugs list. That's what was used for this classification. So that was the response variable that the algorithm was trying to recapitulate. And so this represents a set of simulations that were done for each one of those compounds that was informed by literature data on their in vitro ion channel inhibition. These were simulated across a range of different concentrations from 0 to 500 micromolar, constant concentrations for each. So there are 10 different concentrations selected, and those same concentrations were simulated for each one of the compounds. This was simulated in the cardiac safety simulator assuming a healthy subject. So a healthy female cardiomyocyte is what was used to develop the algorithm. From those simulations, we were able to obtain different metrics that could be used as potential predictor variables for reproducing the cardiac risk stratification from the QT drugs list. So those include electromechanical window, which is the time gap between the end of electric and mechanical systole, as well as action potential and pseudo-ECG based metrics, in particular the QT interval, the QRS interval, and the ratio of the two, which is termed the Index of Cardiac Electrophysiological Balance, or the ICEB. Additionally, some non-standard metrics such as the J-notch to T-peak and the T-peak to T-end were also calculated. And these are important because these have been postulated as potentially being better metrics for assessing torsadogenic potential in the clinic. So using these as the potential predictors, they were linked to the response variable through machine learning approaches. The final algorithm that I'm going to show you is an alternating decision tree. However, other types of decision algorithms were also considered. This was done through a tenfold cross-validation approach, and so the final model represents the average across those runs. Now this was also validated then against 12 additional compounds with known TDP risk. This included moxifloxacin as one of the exemplar compounds. So this is the actual algorithm on the screen. I know it's very small, and that's somewhat on purpose because I'm not going to walk through it in great detail at this point. We've actually submitted this in the form of a manuscript and is currently being reviewed. So we're hoping that it's going to come out in early 2018 in open access form. But just to walk through how this would potentially be used for a novel compound. What you would do is you would do those same simulations based on in vitro ion channel inhibition data or potentially QSAR predicted inhibition data at the same concentrations that were used in the same physiology that was used to develop the algorithm. Then you would walk down the different branches of the decision tree, you add up the scores along the way, and based on the final score, this is converted to a probability of causing torsades de pont, and is converted to a binary classification with a cutoff value of 0.5. So a 50% probability, if it's above that, it's going to be considered torsades positive, below torsades negative. Now, this only requires actually 2 outputs from the cardiac safety simulator. It only relies on the electromechanical window and the ratio of the QT to QRS interval to ICEB as the predictor variables. So those are the only 2 variables that are required as output from the cardiac safety simulator. They're measured at the different concentrations, and so the numbers next to each one of the metrics in the nodes, in the decision nodes, is the concentration rank at which you do the evaluation. Now, as far as how the model actually performed, it was able to correctly classify 85 out of the 96 compounds in the learning set. It did so with high specificity, and being a highly specific algorithm, there is a trade-off in terms of sensitivity. So the sensitivity was 71%. This translates to a false positive rate of 29%. However, when we did area under the receiver operating curve analysis, the area was 0.96, so it was a very robust algorithm in terms of that, discriminating between TDP negative and TDP positive compounds. Now, looking at the validation set, 10 of the 12 were correctly classified, and this included moxifloxacin, which was correctly classified as being torsades positive. We also ran this algorithm on bedaquiline, which was classified as being torsades negative. And this is consistent with its current classification in the QT drugs list at credible-meds.org, where it's considered a possible but not confirmed torsadogenic agent. So there are obviously limitations with this approach because, first off, it's considering monotherapy. And we're really interested in looking at combination therapy ultimately. However, this is reflecting its placement in the development paradigm. This is intended for use in the assessment early on of candidate compounds and potentially where you're more interested in characterizing the compound itself rather than its overall role within a regimen. Additionally, it does not explicitly call in the effects of non-drug factors, because all these simulations were done with a healthy subject physiology. Now, you could run the simulations and run the algorithm by looking at different physiological conditions, such as hypokalemia, for example, to take a drug that potentially is not proarrhythmic based on a healthy subject that could potentially be converted to proarrhythmic based on that change in physiology. This is somewhat of an extrapolation beyond the range of the data, but it is a way of testing potential what-if scenarios for your various compounds. Another consideration with this algorithm is that it converts an estimated probability into a binary classification. But what we saw with this conversion was that drugs that were misclassified for TDP positive or negative were actually within the range of 0.34 to 0.54 as far as the estimated probability. So this suggests that there may be a middle range of compounds or middle range of probabilities where the binary classification may not work quite as well. So at this point, for use of the algorithm, we're considering that the overall classification as well as the estimated probability be considered together when making decisions. As I mentioned, this is submitted in the form of a manuscript. It's currently being reviewed. You can see the working title on the screen. We're expecting this to come out in early 2018. However, in the interim, if there are sponsors and researchers that want to use the algorithm or want more information on it, we would be happy to share this and would be happy to facilitate a discussion. So please contact me or any of my colleagues at CPATH. We'd be happy to make that happen. One other point is that, as with any quantitative drug development platform that the CPATH works on, we intend to update this with emerging data. So this is essentially the first version of the risk stratification algorithm. However, as more compounds become available, we could potentially rerun the model with a greater data set, or if some of the classifications on the learning set compounds change, we would update the model. And additionally, we're looking at incorporating other factors. For example, looking at such an algorithm in combination regimens, as well as potentially different classification schemes, such as a trinary classification rather than a binary. So rather than a plus or minus, perhaps a low, moderate, and high risk type of a classification scheme. So Coming back to the overall QST framework, so this represents the assessment for early in discovery. And really it only relies on in vitro ion channel inhibition data and does not require any clinical data to run this algorithm. However, we want to leverage the data that's obtained throughout the drug development process in cardiac risk assessment. And so for late stage assessment, we're considering a more of a mechanistic or semi-mechanistic PK/PD approach, specifically a physiologically based PK modeling approach linked to the pseudo ECG outputs obtained from the cardiac safety simulator. So what this would do is it would build upon the in vitro ion channel inhibition data to include PK data obtained from clinical trials, ECG data potentially to help verify the model, as well as different physiologies from a virtual population that could be used to test different scenarios. Additionally, through this approach, multiple drugs could be simulated simultaneously in terms of not only their effects on the cardiac ion channels, but also potential pharmacokinetic drug interactions. So this is the paradigm we're looking at for a later assessment of torsadogenic potential. And the idea here is to run a range of simulations in order to identify high-risk scenarios to potentially avoid, as well as identify a safe space, if you will, where the drugs could be used in combinations where it's unlikely that torsades will occur. This work is in progress. We have done a case study of monotherapy with moxifloxacin, when this is in late draft form in the form of a manuscript. We expect that will also be coming out in early 2018, so stay tuned for that. So just to sum up, CPTR through the Modeling Simulation Working Group has developed a cardiac risk stratification algorithm. This is intended for use in early development and is intended to help predict the likelihood of a drug being torsades positive when used in the clinic. It's built on the SimSip cardiac safety simulator, and it's able to convert in vitro ion channel inhibition data into a clinical outcome without requiring any clinical data. This will continue to be updated and will be incorporated into the overall quantitative systems toxicity platform that we're proposing for risk assessment in TB, along with the later stage assessments through mechanistic PK/PD modeling. So with that, I want to acknowledge everyone on the screen, my colleagues at CPATH, and in particular, Sebastian Polak at SimSip, who did the bulk of the hands-on work here. So I have the privilege of talking about the work that he did and then trying to answer the questions. So keep that in mind if you have any very technical questions. And so with that, I'd be happy to answer those at this time.